All right, membrane structure and function. So the cell membrane is, again, surrounded the cell. It's what defines the cell from the rest of the world. And so it is defining the boundary of the cell itself. And again, the stuff on the inside is called cytoplasm. And the membrane is going to be separating the cell from its environment. If it's a single-celled organism, that's like the environment. But if it's a cell within your body, it's going to be from the intercellular space between all of the cells. And so there needs to be some transport, there needs to be movement in and out of the cell. The cells might make things that the rest of the body needs, so it's got to be moved out. There may be things outside the body that the cells need, like glucose and oxygen, so they've got to be moved in. So there's this constant movement of material in and out of the cell, and it is all going through the cell membrane. So there are many different parts to the cell membrane that are going to be integral to this movement and transport, but there's other roles that the cell membrane plays as well, such as like recognition. And so we have the molecules on the outsides of the cells that will recognize one cell versus another, as well as other things within the body. So a very, very complicated system makes up the cell membrane. It's known as the fluid mosaic model in that don't think of it as this sort of static barrier between the inside and the outside of the cell, but instead think of it, it's more like this made up of all these little pieces that continually move through it. So that idea of fluid is really important. It is not this sort of solid barrier. It is more like a fluid itself around the cell, made up of all sorts of little pieces that stay together as the membrane itself, but it's in constant motion. There is the main part of it, so we have this phospholipid bilayer, but then there's going to be lots and lots of these protein molecules and carbohydrates, all embedded or sticking out of or crossing that bilayer as well. So very, very complicated structures found within this cell membrane. Now, remember these phospholipids that we'll talk a little bit about, they have hydrophilic as well as hydrophobic ends to it. So because of this structure, hydrophilic or larger molecules, they can't get through the membrane on their own because there's this whole hydrophobic part to the cell membrane itself. And this is where we're going to have certain channels that will allow transport of certain molecules across the cell membrane. Another little picture of it here, and again at the surface you're going to see all sorts of different things sticking out of the cell membrane. You're going to see all of the little bits that make up the phospholipid bilayer, hydrophobic area in between, hydrophilic on both sides of that as well, and then again we have all sorts of proteins as well sticking on either side of this phospholipid bilayer as well. Membrane carbohydrates, so these are saccharides, oligo, meaning there's a bunch of them in a row there, but they're carbohydrates, and they are bonded to proteins or lipids, and so we have glycoproteins or glycolipids depending on which one they're bonded to, and they're important for the recognition that cells use to essentially communicate with other cells, as well as be able to sort of tell other cells what they're like. And we can see this as an example with our blood type systems. You may know what blood type you have, whether you're A or B or O or AB. That is going to be based on these signals on the cell membranes have on them. So if you are known to be type A blood, what that means is that your blood cells themselves have little A markers on them, not letter markers, but just a particular structure that we have on the outside of these cell membranes that we have labeled A. There's another version of that marker that we have labeled B. And so if you're type B, you may have that marker. And then some people have both markers. And other people have no markers at all. And so depending on which type you have, if you're type O, that means you don't have either one of these markers. It's not you don't have any markers, it's just you don't have these particular two types of them. And then with that in your body, you're going to have antibodies as well. And so these are essentially things that will go around and they look for things to bind onto. And so if you, simplistically speaking here, if you are type A, your antibodies are looking for things that aren't type A to recognize them, whether they are supposed to be there or not. So type A is supposed to be there, so they will actually recognize type B. So someone who's type A blood cannot get a transfusion of type B because their antibodies will react with it. And the opposite is true. If you're type B, you have type A antibodies, so you can't take type A blood if you're type B. If you're type AB, what that means is you don't have any antibodies. So you can actually, you're the universal recipient of blood. You can take type A or type B blood because you don't actually have those antibodies that are looking for them. And then type O, since it has none of these markers, neither the A nor B markers, it actually has both A and B antibodies in it. So they can only receive type O blood. But their blood itself doesn't have any markers, so everyone can actually get type O blood because it doesn't have any markers on it. So type O blood is known as the universal donor. So a little more detail. There's a zoom in of the phospholipid. This is the bilayer that makes up the bulk of the cell membrane. We have, again, you have this outside part of the cell, and then you have the inside part of the cell, and this is the barrier between the two. It is not the only part of the cell membrane, but it is making up a large portion of it. We have this hydrophobic area. So in the middle between the two phospholipid layers, the two of them making up the bilayer, there's this hydrophobic area. So it wants to be away from the water. Now the extracellular environment is aqueous, and the intracellular environment is aqueous. So the hydrophilic parts are going to be attracted to the inside and the outside of the cell. And so this lipid bilayer forms, separating the inside from the outside of the cell. It is made up of glycerol with 2 fatty acid chains to it, and then it has this hydrophilic polar head group to it, which is going to be facing the outside. And so each one of these little molecules here has a hydrophilic end. That's this end up here with the polar head group, and then the hydrophobic tails stick into the middle, trying to get away from the intercellular space as well as the extracellular space, and forming the phospholipid bilayer. There's not the only thing in there. So there are these things called sterols, things like cholesterol. You've heard that being used before. So that is one of the things that you'd find in this phospholipid bilayer embedded in there. And depending on which ones you have and then the amount of them, they actually can change the fluidity of the cell membrane, which is important because you don't want your cell membrane to be too fluid, because then it's essentially going to fall apart. It can't be moving around too much. The things won't be staying embedded in it, and it won't be stable. But I also don't want it too rigid that you can't actually have the cells moving themselves. And so there's this balance between too fluid and too rigid, and that is going to be maintained by these other structures that are in the cell membrane, such as the sterols, as well as the fatty acid tails. Their saturation can be changed around, as we see with saturated fats and unsaturated fats, and that can also affect fluidity. So the roles of the proteins that we see in there, because again, it's not just the phospholipid bilayer, but it's stacked full of these proteins. Some of them are involved in transport, lots of things going in and out. Other ones are involved in reactions. So there's certain reactions that have to be carried out inside the cell, and certain reactions that have to be carried out outside the cell. And so they can actually sort of sit in the membrane and carry out these reactions because they themselves are enzymes. Signaling is important. Again, your cells want to be able to recognize which cells are which, and they have to be able to communicate. And so they do that with these structures on the outside, proteins that will signal that to whatever happens to be recognizing it. Attachment, a lot of cells are going to be connected together or attached to something else. And so this is done as part of the role of the membrane proteins as well. And then similar to signaling is cell-to-cell recognition. So say when your red blood cells are looking for certain things, they're able to recognize your own cells as yours, and they could recognize other cells as not being yours. 2 different terms we can use. So we can say integral, which are ones that are within the actual phospholipid bilayer. They could be crossing the phospholipid bilayer or just sort of stuck in it. And then we also have peripheral ones that are on one side of the phospholipid bilayer. So an example of a particular protein that is found within the phospholipid bilayer is this transport protein here. And this one is associated with cystic fibrosis because that is the disease that occurs when this protein is functioning differently. So if there is a particular deletion, and it's not a huge one, it's only 3 nucleotides in your DNA, it can actually lead to the building of this protein, because remember, protein is built from amino acids, which are coded for in your DNA. So if your DNA is different, the protein sequence might be different, and therefore the amino acid sequence might be different, and therefore the protein might be slightly different. So this particular one changes this transport protein, and CFTR is a cystic fibrosis transmembrane conductance regulator, but this is a protein that allows ions to be transported through your cell. So its job is to move ions out of cells. And things like lungs and intestinal cells, as well as sweat glands, they are moving ions out, and what follows is water. And so on the lining of your lungs, ions get pumped out, and then water is going to flow out as well, because we want there to be mucus on the outside of our lungs, or I mean the inside, but the outside of these cells here. Inside of our lungs, we want there to be mucus layer, and that's made by pumping chloride ions out of the cell, in which case water is also going to flow, and you end up with this wonderful mucousy layer on your lungs, which is good for things like trapping bacteria. Now, this particular genetic change leads to an individual who may, and it's a recessive trait, who may get this particular trait where their proteins aren't pumping out as many chloride ions as would be normal. And so the mucus that is then produced doesn't have as much ions in it, and therefore doesn't have enough water in it. The water doesn't flow out of the cell as much, and so the mucus itself becomes thick, and this leads to the all of the properties of cystic fibrosis. Now, what's interesting about this as well is that those that have that particular deletion of the amino acid are definitely going to be affected negatively through the traits of cystic fibrosis. But those who are only carrying it or not expressing it to the same extent, they may have slightly less thick mucus in their lungs. And so what may happen is that certain diseases like cholera, where you have the cholera bacteria produce a toxin that basically causes those channels to stay open, which causes too many ions to move out, and then that can lead to dehydration. And so this is a current problem. This is not an old problem. This is a current problem that affects a lot of people. And so this particular disease, one of the defenses that we have evolved as a species over time is that if you have a slight reduction in the pumping of those chloride ions, you have some resistance to the devastating effects of cholera. And so this may be why that particular trait is passed down within our population, because in certain circumstances, that trait, though in too great a quantity, when you get both versions of that particular gene allele, are devastating. Having one version may actually protect you from certain diseases like cholera.